Welcome to Labster. In this lab, you will learn the basics of DNA, RNA, amino acids, and protein synthesis. Using the lab pad is an important way to get the most out of this simulation. Is this the first time you've played a Labster simulation? Perfect, then let's get started with your first mission. Let's kickstart today by watching a short movie about protein synthesis.
What you are about to see is DNA's most extraordinary secret. The innermost workings of how a simple code is turned into flesh and blood. This is what Francis Crick called the central dogma of modern biology: how DNA makes protein. It starts with a bundle of factors assembling at the start of a gene. It's these that trigger the first phase of the process, reading off the information that will be needed to make the protein. The gene is the length of DNA stretching away to the left. Everything's ready to roll. 3, 2, 1. The blue molecule racing along the DNA is reading the gene. It's unzipping the double helix and copying one of the two strands. The yellow chain snaking out of the top is a copy of the genetic message, and it's made of a close chemical cousin of DNA called RNA. The building blocks to make the RNA enter through an intake hole. They are matched to the DNA, letter by letter, to make an exact copy of the A's, C's, G's, and T's of the gene. The only difference is that in the RNA copy, the letter T is replaced with a closely related nucleic acid known as U. You are watching this process, called transcription, in real time. It's happening right now in almost every cell in your body.
It's a bit like taking the information off a hard disk on a computer and put it into memory, making it a real program that's running. So that process of DNA to RNA is like running a program, double-clicking on an icon.
When the RNA copy is complete, it snakes away from the nucleus and into the outer part of the cell. Then, in a dazzling display of choreography, all the components of another molecular machine lock together around the RNA to form a miniature factory called a ribosome. It translates the genetic information in the RNA into a string of amino acids that will become a protein. Special transfer molecules, the green triangles, bring each amino acid to the ribosome. The amino acids are the small red tips attached to the transfer molecules. There are different transfer molecules for each of the 20 amino acids. They all carry a specific 3-letter code that will be read by the machine. Now we come to the heart of the process. Inside the ribosome, the RNA is pulled through like a tape. The code for each amino acid is read off, 3 letters at a time, and matched to 3 corresponding letters on the transfer molecule. When the right transfer molecule plugs in, the amino acid it carries is added to the growing protein chain. Again, you are watching this in real time. And after a few seconds, the assembled protein starts to emerge from the ribosome. Ribosomes can make any kind of protein. It just depends what genetic message you feed in on the RNA. In this case, the end product is hemoglobin.
DNA is the molecule of heredity that contains instructions to build proteins. In 1958, Francis Crick described the central dogma of molecular biology explaining the process of gene expression. What do A and B represent in the image? No, this is incorrect. Please try again. That's correct. In 1958, Francis Crick described the gene expression process as the central dogma of molecular biology, where DNA codes for RNA, which in turn codes for proteins. The process whereby the information contained in DNA is copied to mRNA is called transcription. You can observe the main differences between DNA and RNA by clicking on the View Image button. Which statement is true? That's correct. The DNA base T is switched for U in RNA. The sugar molecule that makes RNA is called ribose and has a hydroxyl group attached to its second carbon. DNA, on the other hand, is missing this hydroxyl group and is therefore called deoxyribose nucleic acid. This hydroxyl chemical group makes RNA molecules more reactive than DNA molecules. The mRNA strand is synthesized in the 5' to 3' direction. The circles in the image indicate the chemical group in which the next base is attached. Which image has the circle in the correct position? You are right. The next base of mRNA is attached to the hydroxyl group on the third carbon of the ribose sugar. DNA and mRNA synthesis proceeds in the 5' to 3' direction. What does 5' and 3' mean? Genius. 5' and 3', or 5' and 3', refer to the position of carbon atoms in the sugar backbone. The 5' carbon attaches to a phosphate group and the 3' carbon attaches to a hydroxyl group. Protein synthesis is performed by an RNA protein complex called the ribosome. As you can see in the image, a ribosome is comprised of 3 sites called E, P and A. To which site does tRNA add its amino acid to the polypeptide chain? You are very good at this. Protein synthesis occurs as a result of the collaboration between tRNA, mRNA and the ribosome. tRNA molecules traverse 3 sites of the ribosome in the following order: A, P, and E. tRNA molecules add their amino acids to the growing polypeptide chain at the P site. The A site is where tRNA molecules, with their attached amino acid, bind to the mRNA codon. The E site is where tRNA molecules reside before being released from the ribosome. Now let's watch an animation. Genetic information is stored as DNA and copied as messenger RNA during transcription. In mRNA, genetic information is encoded in 3-letter units, codons, made up of the bases uracil, cytosine, adenine, and guanine. In the ribosome, each codon is translated into one amino acid. Amino acids are linked by covalent peptide bonds. The formation of a peptide bond produces one water molecule. Amino acids are linked by a specific type of covalent bond. The reaction of this bond produces one water molecule. This bond links multiple amino acids together. What is it called? Smart. A covalent peptide bond links 2 amino acids, thereby forming a long chain. Peptide bonds occur between amino groups and carboxyl groups of neighboring amino acids. The process of making a peptide bond produces one molecule of water. An amino acid molecule consists of 3 main components: a carboxyl group, an amino group and a side chain symbolized with an R. A peptide bond occurs between the amino and carboxyl groups of neighboring amino acids. A sequence of amino acids, called a polypeptide, grows from the N-terminus to the C-terminus. This polypeptide chain represents the primary structure of a protein. A sequence of amino acids, or a polypeptide, grows from the N-terminus to the C-terminus. This polypeptide chain represents the primary structure of a protein. The linear sequence of amino acids within a protein is considered the structure of the protein. Nice. The primary structure of a protein is the linear sequence of amino acids. Which level of protein structure is formed by the weak bonds between oxygen and hydrogen atoms within the polypeptide backbone? Smart. The oxygen and hydrogen atoms within the polypeptide backbone can form hydrogen bonds. These weak bonds result in the local folded structure known as the secondary structure. Two basic types of protein secondary structures are alpha helix and beta sheet. The alpha helix is a right-handed coil. The beta sheet can form two or more segments of the polypeptide chain lying side by side. The entire folded protein 3D structure is known as tertiary structure. The main two secondary structures of proteins are alpha helix and beta sheet, which are represented respectively by which two letters in the image? Perfect. The amino group and carboxyl groups in the neighboring regions of the protein chain can cause certain patterns of folding to occur. The alpha helix looks like a coiled spring. The beta sheet looks like a protein chain that is folded to lie alongside itself in a pleated arrangement. Proteins are modified in many ways after translation. For example, the hormone erythropoietin undergoes post-translation modification, called glycosylation, before it can be considered biologically active. Glycosylation is the addition of sugars. EPO has 4 linked sugars. Now, let's finish by answering a few more questions about DNA and RNA. Click the View Image button to view a eukaryotic cell. Where do eukaryotic cells synthesize proteins and which number represents this organelle in the image? You are absolutely right. In eukaryotic organisms, transcription takes place in the nucleus. The resulting mRNA is then transported to the ribosomes in the cytoplasm where translation occurs. What is the name of each sequence of 3 nitrogen-containing bases of RNA that codes for 1 amino acid? That's correct. The mRNA nucleotide triplets are called codons. The codons are written in the 5' to 3' direction. Each codon encodes 1 amino acid. The tRNA shown in the diagram will be able to form a base pair interaction with which mRNA? Bravo! The interaction between a codon and anticodon will always be antiparallel. The anticodon 5' ACU 3' will bind to 3' UGA, which is the same as 5' AGU. 3 types of RNA cooperate to perform translation. A. Read codons and deliver the amino acids. B. Carry the genetic information from DNA. C. Catalyze the assembly of polypeptide chains. Which type of RNA matches the A, B, and C descriptions, respectively? Perfect. There are 3 types of RNA that cooperate to perform translation. Those RNAs are messenger RNA or mRNA, transfer RNA, or tRNA, and ribosomal RNA, or rRNA. Well done. You have completed the simulation. Let me ask you one last question. Do you feel like you gained relevant knowledge during this simulation? Thank you for your feedback. Your score has been saved together with your 100% progress. Congratulations, you have completed the simulation. We hope you enjoyed it and you feel wiser now.